Thanks for that introduction, Craig. And as you said, my name is Matthew Crawford. I'm a senior scientist at Rouse Technologies and a consulting scientist at Nanomicrotech, specializing in mass spectrometry and chromatography separation of biomolecules, peptides, small molecules, and conjugates. So I'll be talking today about the unique challenges and opportunities that face us in peptide, incretin, hormone separations research. So the first question we might ask ourselves is, what is GLP-1 and why do we care about purifying it? So GLP-1, as you can see here, if I can get the pointer up, is a 33 amino acid peptide that interacts with a receptor that's embedded in the cell membrane of different cell tissues in your body. So it helps to regulate glycemic control, and modifying it for different PK/PD parameters renders a few different off-target effects, some of which are desirable, and we can leverage for therapeutic purposes. So back when I first started working with these compounds in undergraduate research, these were quite a niche peptide. They were used for treatment of type 2 diabetes mellitus. But as you can see, and anyone who's been watching the news has noticed, the market for them has expanded greatly now that they've been found to be effective weight loss drugs. That being said, there are quite a few other modalities that this can help with, a few different disease states. So Alzheimer's, Parkinson's, it's got quite a few neuroprotective effects. So it's even being used to pre-treat for CTE, which is the concussion injury that American football players and people who get repeated head injuries can suffer from. So it really does have quite a few uses. And right now we're in the middle of what are called the GLP-1 wars, mostly being fought over by Eli Lilly Company and Novo Nordisk. Actually, back when I pulled this information, Lilly was on the smaller side, but they've actually currently taken a larger share in the market. I thought about updating this image, but I thought it really drives home the point that this market is changing extremely rapidly, and so we as purification specialists need to be ready to adapt to the changing needs of the market. So GLP-1s that have been modified quite a lot over the years from their native forms. Pharmacokinetics and pharmacodynamics are the main drivers of peptide design. So there are two major routes that are causing issues with the effectiveness of this drug. So one is a peptide protease in your blood called DPP4, dipeptidyl peptidase 4. It cleaves these N-terminal histidine alanine residues in the peptide and render it about 1 million-fold less effective at the receptor. So that issue has mainly been solved through the substitution of an AIB residue in place of the alanine here, which prevents the protease from actually cleaving it. The other issue is renal clearance. So your body tries to get molecules like this out of your blood. In the native GLP-1, it has a half-life of about 30 seconds, which is just not long enough for an effective drug. So a lot of modifications are used to try and remove it from large levels in circulation and kind of store it in a few other areas, such as proteins or in certain tissues, where it can then be slowly re-released and have a longer-term effect. So due to these various clearance issues, there have been quite a few different approaches that have been used by scientists in order to try and improve the half-life of this compound. So exenatide was one of the earlier versions of GLP-1. It was taken from the American Gila monster in the Mojave Desert in the Southwest. It has a major sequence dissimilarity from our native GLP-1, so that's how it has increased proteolytic resistance and also improved renal clearance rates, but it's still a twice daily injection. Liraglutide was really one of the blockbuster drugs. It uses a lipid moiety to help embed in the serum albumin in your blood. And while it's in that protein, it's not exposed to the DPP4 or renal clearance that's going to cause issues when it's trying to be effective. Semaglutide follows a similar train of thought, using a lipid in order to help embed an albumin. It's a bit more effective at embedding, and so it's a once weekly injection. So I won't go into all of the different types here, but if you look, you can see that there are quite a few different ways of approaching this. But of specific note is at the bottom here, orforglipron. That is a new drug that's being produced by Lilly. It's in phase 3 clinical trials, and it's a small molecule peptidomimetic. So small molecules have historically been a lot easier to produce and distribute, as well as being orally bioavailable. So I would keep your eye on this molecule, as it's potentially going to change the market quite a lot. So now that we've established why we care about GLP-1 analogs, why don't we get into some of the early discovery and downstream process controls that are being used to actually generate these peptides at high quality. So what is really interesting about this is that there are a few similarities between discovery, research, and development, as well as process purification, but there are also several key differences that have historically kept them quite separated, both temporally, so they're happening at different times. So discovery is happening a few years before a lot of the process and manufacturing, They're also separated kind of spatially and organizationally. So you'll sometimes have your research group in one building and your manufacturing group in another building, and they're just often not allowed to talk to one another, or even institutionally. So they're again, they have very different cultures and how they operate, and so they're often not talking to one another and not able to pull data across the various modes. So you can see here, like in research and discovery, we're looking at microgram to low gram scale, whereas in process and manufacturing, we're going up to the kilo scale, or often even beyond that. So this is just one example of all the differences that you can find between the different types of purification that scientists are doing. And most of my work actually specializes in R&D, so research and discovery, and usually our main question is how fast can we go? So speed and versatility are really our highest priorities. Purity is a nice to have, and reproducibility is also a nice to have, but they are not our critical metrics. Kind of a more important thing is how fast are we actually able to make this go? So a lot of my work at Rylus Technologies is focused on discovery research, as I described. And so we actually have developed a process now for purifying compounds in the library scale. So 96 well plates, 24 well plates, 48 well plates. So a lot of scientists are generating peptide libraries on synthesizers, automated synthesizers, and the purification has really struggled to keep up, as you can generate a 96 well plate in a matter of a day or two, but purification, you can often go only 6 to 10 compounds a day, so it can really take up to 2 weeks to purify an entire 96 well plate. So the process that we've designed here is a 3-phase process. So phase 1 is running a high-resolution peptide QC method on the peptides in order to determine what their actual starting purity is. Phase 2.1 is running a trifluoroacetic acid-based pre-QC to scout the actual focused gradient that we're going to use on reverse phase in order to separate the peptides. Phase 2.2 is a trifluoroacetic acid-based mass-directed prep on the peptide library. So that would be on the low milligram scale usually, so from 1 to about 10 milligrams, maybe up to 20. And then we do a final QC of the peptide after we've purified it to show the improvement over what was done before. So in order to demonstrate the capabilities of this process, we wanted to design a high throughput peptide purification experiment. What we found is that solid phase extraction, though it is highly automatable, often does not achieve the purity required. So you can get a crude purity of as low as about 20%. The SPE can get you up to about 50. But in a peptide context, the closely eluting contaminants that SPE does not remove are actually the most likely to be competitive inhibitors at the receptor site. So we really need a process that would allow us to purify 96 peptides per day on the low milligram scale. We want to use a single stationary phase that's highly scalable, so we can do hit validation and then do a very rapid scale up without having to revalidate the chromatography resin that's being used. And we wanted to use mass spectrometry in order to increase the robustness of the purification process. So we received a peptide alanine walk from a high throughput synthesis group, as I described. It was actually a liraglutide alanine walk without the fatty acid moiety on it. We received in a 96 well plate format and returned in a 96 well plate format, which integrates quite nicely with their sample tracking. So they're able to maintain speed in their sample tracking databases. And our goal was to achieve a high throughput process using these nanomicro resins. So this is what we actually see in our crude QC runs. So as you can see, we have a large DMSO slug here that's coming through in the early phase around here. And then we run a quite long gradient in terms of column volume. So it's quite a thin column, just a 2.1 by 150, very low column volume, but it's a 5 to 95% B over 10-minute gradient. I believe it's about 30 column volume, so it's a fairly long gradient. And that helps us to really pull apart all these little closely eluting contaminants that we might not be able to see through other processes, and allows us to make sure that we're getting a true measure of the input purity of the compound that we're working with. These peptides were received, again, as a crude cleavage mixture off of resin, which, there's quite a few different things in there. I believe it was passed through an ether crash-out precipitation, which did remove some really far away contaminants, but it didn't actually kind of improve the purity of this compound up to the level where it could be reliably tested with. So once we have the crude QC, we know exactly what purity we're starting with. We then move on to the prep system. So we found that UV alone at 215 and 280 is insufficient to identify the target, as it's often not the major component. So we want to have something online that allows us to properly identify the compounds that we're selecting for separation out from the compound mixture. So mass spectrometry is included in both the analytical and preparative runs to ensure proper collection of the target. EIC, which is an extracted ion chromatogram, allows us to properly identify the target. The extracted ion chromatogram is made using a predictive system that we've designed where you can calculate the M over Z values for higher charge states and use that as an and measurement, instead of an or measurement, it's an and measurement with UV in order to make sure that both criteria are satisfied before collection of the target is performed. And we use the best ion from the analytical pre-QC for the preparative runs. That's not necessarily the most abundant, it's more the most robust ion, so the one that's going to change the least between the analytical and the preparative runs. And so once we perform the preparative run, we'll then run a final QC on the compound. So here we're using an Agilent Advanced Biopeptide Mapping column. Again, the same mobile phase as the pre-QC, the same gradient as the pre-QC. And you can see, well, we've still got a couple of small early eluting peaks here. The vast majority of this compound is now target. And again, that is confirmed by mass spectrometry, as we'll see on the following slide. So what we can see here is the crude mass spectrum of the compound that was coming off of our pre-QC, high resolution pre-QC method. What's interesting about this is despite the high resolution separation, we're still seeing a bit of coelution with a contaminant here. So our starting material has, or our target has a molecular weight of 3,751.2 Daltons. That is its molecular weight, not its exact mass. And what we're looking for here is a charge series. So we're going to use the M over Z equation, calculating Thomson's, of a +2 charge state of 1876, which we do not see in the mass spectrum, but we do see the 1251.4 plus 3 compound, ion rather. We see the 938.7 and the 751.2, and then we can also see here there's actually a contaminant material that's a little bit higher molecular weight. So what that can be is polycoupling, for example. So if you're trying to couple a single histidine residue, you might actually couple two if you don't fully wash out your activated amino acid coupling solution. And that's the kind of contaminant that can actually cause a big problem in peptides and discovery, because something that's sequenced similar like that will have a similar affinity for the receptor and might not actually activate it. So you might be including an active inhibitor of your compound in your separation. So once we actually run the mass spectrometry on the purified product, we can see here that now we've removed that heavier molecular weight contaminant. We've actually clarified the compound and enriched it enough where we're picking up an additional charge state. That would be the +6 up at down at 626. And what's really nice about this is we wouldn't necessarily be able to see this through UV if the method wasn't very, very high resolution. And in discovery, often we don't have time to be running several hour methods. So it is nice to be able to use an orthogonal method that takes a shorter amount of time in order to confirm we've removed other contaminant issues. And so here are the tabulated results from the 33 compound alanine walk. So it's, again, a liraglutide alanine walk. The crude purities that we received from the manufacturer averaged about 45% with large deviations in purity, so a relative standard deviation of 22.4%, which means you'll often be getting stuff as low as 20% or as high as 70 or 80%. And so it's going to really throw off your potency calculations when you're running this in a bioassay. So after purification, we averaged 88.34%. We were targeting 85%, so this was considered a success with a much smaller deviation. So it's 4.5% relative standard deviation versus the 22%. What's interesting here is we have the total process time of pre-QC, prep, and final QC total about 18 hours, which only 2 technician hours. So it's a highly automated process that can also be stacked. So you can be running a pre-QC while you're also running a prep and a final QC as long as you have the instrument availability. We did see 2 failures in this process. So up here we have a synthetic failure. So it was only 24% coming in, 27% coming out. So that was actually a failed synthesis. They didn't make the target. And then we had an instrument failure here, which was a power outage that we had on the instrument. So we're stabilizing that. We're working on improving the robustness of the process. But as always in discovery, it's a heavy work in progress. But we're pleased with the results we got out of this. So something else that is quite critical in discovery research is adaptability to other kinds of materials. So unlike in a lot of process methods, we need to use this across a very broad range of compounds. So here we ran it against 100 compounds from each of 4 classes, the same process. So that would be small molecules, 12 mer peptides, 30 mer peptides, which was GLP-1s, and then 55 mer peptides, which is starting to get up to the synthetic protein scale. And as you can see, the crude purity drops off quite heavily here over the course of the increasing size of the molecule. And we do see a drop off in our compounds as well, but in purified, but it's much less pronounced, and this will enable a much higher confidence moving forward for groups that are using this in bioassays. So once we have these purified compounds, we then need to pass it through a few different tests, whether it be a receptor activity assay, a solubility assay, biomimetic chromatography for membrane or albumin binding, kind of establishing several PK/PD parameters that will then inform the next library synthesis. So this rapid turnaround is really useful for our clients in terms of refining their peptide synthesis libraries. So now that we're moving out of the discovery stage and let's say we identify a hit that we want to then get into larger scale, we need to start developing a process method. So here the target is extremely high purity and high levels of reproducibility. Versatility is a lot less of a priority, and yield also becomes a much higher priority as we are starting to run into kind of cost issues. So there's a lot less cost sensitivity in the discovery space than there tends to be in the process space. So often when moving from the discovery to the process phase, the process group will then redevelop the entire separation from scratch. The pro of this is that the process group has full control over their purification. They have a very good system for generating high quality material. But a con is that they can't use any information that was gathered from the discovery groups. They might have noticed some very high quality separation technique that the discovery group might not even hear about. And so, for example, here, you can't really use TFA in the later stages of process because it's toxic, whereas discovery groups love to use it because it's a really nice universal unpairing agent and modifier that yields really nice sharp peaks and tends to give you a higher purity, but you just can't use it for stuff that's going into animals as readily. So when doing that process development, again, they're taking it from scratch and it's taken on a more case-by-case basis. So you're going to be optimizing the separation of a single compound. You're not going to be getting a 96-well plate of libraries. You're instead trying to get very high purity, very stringent quality metrics, perhaps even going into GMP processes, where you're going to be trying to generate a really, really high purity compound, and that you're going to be generating the exact same compound every time. Orthogonality of methods is also appreciated as in discovery, we can use a wide range reverse phase capture and then a focused reverse phase polish if required. Whereas in process, you really need to use completely orthogonal methods. So reverse phase and ion exchange, or Hillick and SEC, the various kinds of truly orthogonal methods that will pull apart things that might be missed under things, separation modalities that separate by a similar metric. So when you're doing this process development, there are quite a few considerations you have to take as you're performing the scale up, and it kind of needs to go in a stepwise process. So first you need to identify your target purity. So are you going into small animal studies? Are you going into large animal studies? Is this going into humans for clinical trials? Is this now an FDA-approved drug or going to be an FDA-approved drug? So you need to really identify your target purity, as these last few percent would be nice to get, but it really is a logarithmic process in that it gets a lot harder to create a higher purity compound than it is a low purity. Going that last little 0.5% is a lot harder than the first 50%. The next consideration would be capacity. So how much material are we going to be able to load onto this column? How big of a column and how much media will we need? Another consideration is the yield. So when you're performing this separation, what is the overall yield, which is heavily dependent on, let's say, the intake purity. Maybe you need to talk to the upstream development people in order to get a higher input purity compound. Then that might save you so much time downstream that it saves you money as well. Then there is a bit of a break here between the compound parameters and the column parameters. So efficiency is how fast are we able to do this separation. Column lifetime is also critical. We want to know how long the media is going to last. And then solvent can also be critical. But these all need to be considered together. So if you can use acetonitrile, but then reduce the process from 200 minutes down to 60, even though acetonitrile is more expensive, if you use less of it because of a shorter process, then you're going to end up with a cheaper overall process. A similar thing if you use, say, methanol, but the increased viscosity causes you to have fewer cycle lives on your column. If you need to purchase more and more and more resin, the cost saving of using methanol might end up being a little bit less than you would save if you just went with a less viscous solvent. And so this is where the nanomicro resins really tend to shine. So like I was saying earlier, this is a fully monodispersed, but also highly scalable resin. So we're able to use this both in the laboratory setting in sub-milliliter columns, all the way up to the ton scale for manufacturing. So you can see here the normal spray dry and sol gel process leads to a fairly irregular column packing. And so there's actually quite a polydispersed set here. So these are 3 different manufacturers. You can see that it's quite a wide distribution, whereas the Nanomicro's patented process generates a very homogeneous output media with very uniform beads. And that enables us to ensure that we're going to have very reproducible runs when we're running this. And again, it being a scalable resin allows us to move from the discovery to the process quite quickly without having to select a new media. So once you kind of outline your criteria, you then need to outline your process steps. So there's almost always going to be 2 to 3 chromatography steps in any kind of process or manufacturing. So a single one is usually insufficient. There are 2 main modes of generating GLP-1s at this point. There's the synthetic route and there's the biological route. And they will have different purification schemes, as a cell is going to have quite a few protein contaminants. It's going to have other peptides that are just completely unrelated to yours but are used by the cell. Whereas the synthetic route is going to have no proteins in it, but it's going to have a lot more sequence similar peptides, as they're just short truncations of the material that you're trying to work with. So that's going to influence what you select for your resin. So let's say you're purifying this R protein here, and you want to go to this peptide. You might want to use hydrophobic interaction chromatography. But if you have a bunch of proteins that stick to the column, you're going to want to avoid that. And then you can move on to your actual crude peptide once you cleave it out of the protein. And you're going to want to use an orthogonal method. So you might have like PBRP here, or move on to Hillic to get the final polish and really achieve that high purity. Whereas for synthetic, again, you're not going to have proteins in there, so you're much more likely to be able to use something like a C18 resin without fouling it. Proteins really like to irreversibly bind to a C18 resin. And so you're going to get a different crude purity, but you're not going to have the same contaminant profile. So you're going to want to select a different set of purification strategies in order to move forward with those. And so we wanted to take this same nanomicro resin that we were using in the discovery phase and actually try and develop a process method for it. So one thing here that's critical is ensuring that we have compatible mobile phase modifiers with our instruments. So our instruments are stainless steel, which are not super amenable to sulfuric acid, hydrochloric acid. So I adapted it to a phosphoric acid-based method, which actually helps to extend the lifetime of our compounds and yields a phosphate salt on the GLP-1 when it comes out, which is fully biocompatible. So we don't have to really do a salt exchange at the final steps. We would still want to do, again, potency measurements, all the in-process controls, but the fact that we're able to use such a biocompatible modifier really helps the process. And so in using this resin, we were actually able to do an almost isocratic separation. It was a very, very shallow gradient. And we were targeting greater than 95% purity at the capture step. And we were actually surprised to find that we achieved greater than 99% purity on the capture step. The yield on this wasn't amazing, but what's nice about the process is we can actually take the 2 sides of the peak that weren't passing the criteria and recycle them into the next process in order to help improve yields. We just need to absolutely be sure that we do our in-process controls. What's important to mention here as well is that this cannot be used as a single process step in manufacturing. You do need to have an orthogonal method for separation. And the issue is that you're going to have a lot of regulatory problems when you do try and file with just a single step like this. So what we can conclude from this is that discovery and process methods are highly different, but there are really some attributes of each that can inform the design of the other and improve the efficiency. And so really just, if I can say one thing again and again, it's really just talk to the other members of your team and talk to other teams, make sure that there's lots of communication going on. I know it can get a bit tedious when there's all these emails going back and forth, but you can save so much time and money. The scientists, the engineers, we have different backgrounds. But we're all chromatographers. We're all, trying to achieve a similar goal, and we can learn from each other from these different perspectives. So we just want to bear in mind all aspects of this process when we are, doing our method design, selecting our resins in early discovery. A lot of early discovery scientists, again, they don't consider process when they're making their resin selection, and that it ends up costing the company further down the line. So just, let's just keep talking to one another and make sure that the full process is understood as best as possible by everyone involved. And just to reiterate that point, so here are a few different recommendations and advice for discovery and process. Again, these apply to both in equal measure, but you'll just approach them in slightly different ways. So for example, buffer salt species in your mobile phase A or buffer composition. That's going to affect both discovery and process. So if the discovery person can make it work with a biocompatible mobile phase, That'll help the process guy to be able to, what we like to say, hit the ground running and move as efficiently and fast as possible. Mobile phase B, your organic modifiers. Again, due to our lower cost sensitivity and discovery, we tend to use acetonitrile for a lot of our reverse phase separations because it's a better eluent. But there are processes where we can use methanol, and that would again save time for the redevelopment of the process downstream. So it's not just on the conditions, but also on the media. So selecting a matrix that is, let's say, scalable. That's what's nice about these nanomicro resins, is that they can go all the way from the early discovery scale in just really tiny columns. I mean, 100 microliter columns sometimes, all the way up to the ton scale where you're using dozens of liter columns and just manufacturing this on a huge scale, making sure that that supply chain is robust. So that's another thing that's nice to work with nanomicro. You can be assured that the supply chain is not going to be interrupted. So taking all of that into consideration, I just wanted to say thank you for your time. Thank you for letting me talk about GLP-1s. I could easily go on for quite a lot longer, but I just wanted to make sure that we save some time for questions. And if you performed any process that differs from these principles, I'd love to hear it. I'm not the only authority on GLP-1s. Everybody's doing practical work in the lab that can really inform what I'm doing, how I can improve as a scientist. And so, like I kept saying, communication really is valuable. So once again, thank you for your time, and please let me know if there are any questions.